So we've made resources for reviewing in biology. From our GIF review to our study tips video to our mega biology review video called Stroll Through the Playlist. Our Stroll Through the Playlist was actually our longest video in Amoeba Sisters history. This one won't be quite that long, but it will address an area that the Stroll Through the Playlist couldn't go into very much. A review of how to do general genetic problems. By that, I mean this video will review Mendelian one-trait and two-trait crosses, then non-Mendelian traits like incomplete dominance, codominance, multiple alleles, and sex-linked traits. We'll end with pedigrees. Before starting, I've got 5 things to mention first. Number 1, a sheet of paper will be very useful for this. You might want to pull one out now with a pen or pencil. Just like our Stroll Through the Playlist, when you see Gus pop up, you can pause the video and work out a problem to check how you're doing. Number 2, we'll use some genetic vocabulary here, and we're making the assumption that you've already seen these words before. Number 3, with genetic problems, the symbols used in textbooks, especially with incomplete dominance and codominance, can vary. Some use superscript, some use different letters. It's not the symbol you need to focus on, but rather the concept. But just to note, if you're choosing the letters to write out, you might want to pick letters that have very different looking capital and lowercase letters, unless you go to a lot of effort to make them look different. Number 4, remember that when you're doing a genetic problem, you are determining a probability. But just because a Punnett square tells you that 1 out of 4 guinea pig offspring will be hairless, doesn't mean that there will always be 1 hairless guinea pig every time you have 4 offspring. It can show it's possible, but it's still a probability. Number 5, and this is a big one, please realize the topic of genetics is complex, far more complex than simple gene traits you tend to see in Punnett square problems. Traits can be polygenic, which means many genes control one trait. Some traits can be pleiotropic, which means one gene controls many different traits. We mentioned epistasis in one of our videos, which means that a gene's expression, whether it's expressed or not, can be impacted by the expression of another gene. And we released a video about epigenetics. This involves factors at work that are not part of the DNA sequence, but yet can influence gene expression. Okay, with those 5 things said, let's get started. First, with understanding a Mendelian one-trait cross, which includes monohybrid crosses. Let's assume here we have these 3 guinea pigs. We're using the letter H to represent an allele, and we're assuming the presence of a dominant allele means the guinea pig will have hair. This guinea pig is homozygous dominant, this one is heterozygous, and this one is homozygous recessive. Knowing that, can you complete the genotypes? Big H, big H, little h, and little h. Remember, it only takes 1 dominant allele and it would have hair. Also remember, there are 2 alleles per guinea pig here because each guinea pig gets 1 allele from each of their parents. Please cross a hairless guinea pig with a heterozygous guinea pig and give the phenotype and genotype ratios of the offspring. Notice that I had to figure out the hairless guinea pig's genotype would be little h. I remembered it only takes 1 dominant allele in this example and it would have hair, so it can't have a capital H. I arranged the Punnett square, it doesn't matter which side I put the parents on. You will notice the genotype ratio of the offspring here is 2 big H little h to 2 little h. This can be reduced to a 1-to-1 ratio. The phenotype ratio is 2 with hair to 2 hairless, which can be reduced to 1-to-1. Genotype ratios and phenotype ratios don't necessarily match, although they did in this case. On to a Mendelian two-trait cross, which includes dihybrid crosses. Moving on to a different animal, cats. With our Mendelian two-trait and dihybrid cross video, we mentioned a fictional trait of the love for sinks, because our cat Moo, and many cats we've been told, seem to have an affection for sinks. Again, probably not a genetic trait, but for this example, let's use this fictional trait. We will assume here that the presence of a dominant S allele leads to the trait of a cat loving sinks. Only 2 recessive S alleles would result in the non-sink loving trait. So if we have a cat that is heterozygous for both the traits of having hair and loving sinks, what would that cat's genotype be? Big H, big S, little s. Let's cross it with another cat with that same genotype. Big H, big S, little s. Now, a dihybrid calls for a 16 square box here. What would be the gamete combinations that we put on the top and side of this square? Recall the FOIL method. The gametes along the side would be big H, big S, big H, little s, little h, big S, and little h, little s. Notice how the gametes have one of each allele type, meaning you won't have a big H or a big S in a gamete. You get one of each. Go ahead and fill that dihybrid cross in and give us the phenotype ratio. Here we are. Notice we put the H letters here first, and then the S letters, similar to how the genotype was written for the parent cats. That's a 9:3 to 3:1 phenotype ratio, which, if you cross two organisms that are heterozygous for both traits in a dihybrid cross, you will find that phenotype ratio to occur. But for a two-trait example that doesn't have two heterozygote parents, you can check that out on our full content video. Okay, we're leaving Mendelian genetics now. Mendelian genetics followed an inheritance pattern where having a dominant allele meant the dominant trait was expressed. But in non-Mendelian inheritance, we'll see that's not always how it works. Take incomplete dominance. Keep in mind before starting, there should be clues that a problem is involving incomplete dominance or some other non-Mendelian trait. Incomplete dominant traits tend to have an intermediate phenotype, almost an in-between phenotype. The snapdragon example is a popular one. Here's a red snapdragon with a genotype big R, a white snapdragon with genotype little r. But the big R, little r genotype leads to a pink phenotype. In incomplete dominance, one allele is not completely dominant over the other. What would be the genotype and phenotype ratios of the offspring from two pink snapdragons crossed? 1 big R to 2 big R to 1 little r would be the genotype ratio. 1 red to 2 pink to 1 white would be the phenotype ratio. This is different from codominance. In codominance, both traits are expressed fully. So for codominance, we like using different letters entirely for this reason. In a certain type of chicken, genotype BB results in black chickens, WW results in white chickens, and BW results in black and white speckled chickens. What would be the genotype and phenotype ratios of offspring from 1 black chicken and 1 black and white speckled chicken? 2 BB to 2 BW, reduced to 1:1, would be the genotype ratio. And as for the phenotype ratio, 2 black chickens to 2 black and white speckled chickens, reduced to 1:1. Now it's time for a genetic problem with multiple alleles. Blood types are a great example of this. If we have these 4 blood types, A, B, AB, and O, can you write the genotypes? And just a hint, it's common to write the alleles as exponents on the letter I, although it doesn't have to be written that way. Here they are. Now, if there's one parent that is heterozygous type B and another that is heterozygous type A, what is the percent chance that the baby from these two parents will be type O? So after working this out with the correct genotypes around the Punnett square, it is a 25% chance that the baby will be type O. Keep in mind that blood types can also be positive or negative, which is related to Rh factor, which our video does not go into. Next, sex-linked traits on sex chromosomes. In these Punnett square problems, you are usually told it is a sex-linked trait, and then also given information about whether an individual is male or female. In these problems, you are working on Punnett squares that involve alleles on sex chromosomes. But as we mentioned in our old video, please know that individuals can have more or fewer sex chromosomes than what might be written in a Punnett square. Let's consider the recessive sex-linked disorder hemophilia. If I tell you it's sex-linked recessive and we use the letter H, how would you write the genotype for a male that has the disorder? You'd write XHY. Notice that it's only placed on the X chromosome. Generally, sex-linked traits will be found on the X chromosome, although there are some exceptions. If a male was XHY, this individual would not have the disease since the disease is a recessive sex-linked disorder. Which of these female genotypes would have the disease hemophilia? Only the female genotype with XHH. Remember, the heterozygous genotype, X big H, X little h, still has a dominant allele, represented by the capital H, which means this individual does not have this disorder. If a male with hemophilia and a female who is homozygous dominant decide to have a child together, what % chance is it that the child would have hemophilia? It is a 0% chance. Also, do you notice how the male children receive their X chromosome from the female? They receive their Y chromosome from the male. All right, that's a lot of genetic problems. Now our last topic, pedigrees. Pedigrees can be used to track a trait of interest, and they use many of the concepts we've been reviewing. A reminder that the shaded shape in a pedigree is generally the trait of interest. Some people will also do a half shading to represent the heterozygous genotype, as we mentioned in our pedigree video. However, this isn't always done, and we're going to assume half shadings have not been done in our examples. Take a look at this pedigree. What shape is supposed to represent females? That's right, the circles. The males would be represented as squares. So in this pedigree, assume you are told the shaded shapes represent individuals that have an autosomal recessive trait. Because it is autosomal, the trait is not on a sex chromosome. In our pedigree video, our trait of interest was tracking attached earlobes. But as we mentioned in that video, this trait may be more complex than a single gene trait. We're going to use the letter E here, so any of the shaded shapes must have the genotype e. So if given this pedigree, can you determine the genotypes of the rest of the individuals? Just a reminder with pedigrees, it's often ideal to fill out the genotypes of the shaded shapes first, before determining the others. And in this case, you know the shaded shapes will all be e. So now try and fill out the rest of the genotypes. Here are all the genotypes. A few things to point out. Notice in generation 1, individual 1, the individual must be big E, little e. That's because there is a little e offspring. And so if the individual was big E, that would not be possible. This is the same situation with individual 1 in generation 2. Notice in generation 3, individual 2, this male must be big E, little e. This male cannot be big E because it would not be possible to receive a dominant allele from both parents. All offspring receive one allele from each parent. Notice in generation 2, individual 4, this male could be big E, or big E, little e. We don't know. Even if this individual had 10 children that did not have the trait, you still would not know the genotype for sure. The only way you would know for sure is if they had a child with a genotype e. Because a child with the e genotype would reveal that both of these parents would have to be the heterozygous genotype. But there's not a child represented by a shaded shape here. What if I didn't tell you that this trait was autosomal recessive? Could you show why this pedigree is likely not tracking a sex-linked recessive trait? So, if this was tracking a sex-linked recessive trait, the shaded shapes would then represent genotypes that are sex-linked recessive. Filling out the genotypes of shaded shapes first, here they are. So can you determine why this is not likely? So take a look at parents 1 and 2 in generation 2. We know that generation 2, individual 1, is a male. The individual must have genotype X big E Y, because if the genotype was X little e y, the shape would be shaded. Notice a Punnett square with parents 1 and 2 of generation 2 shows it is not possible to have those offspring genotypes in generation 3 from the pedigree. This specific pedigree is not likely to be tracking a sex-linked recessive trait. So that was a lot to review. What if you're still stuck? Check out the full content videos, which are each under 10 minutes in our genetic series. Practice a lot, and finally, connect this to why it matters. We have some links to check out with some more fascinating real-life examples in our video details so you can discover why gaining an understanding of genetics is such a worthwhile endeavor. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.